in this last lecture, we are going to be looking at how to prevent the loss of excessive water in the urine. And we are going to take a look at some animals that have been well studied, in particular the kangaroo rat, again from the early work by Knut Schmidt-Nielsen already in the 1950s, 1960s. Oops. As a reminder, this is a kidney. In the kidney you have first the outer part, which is the cortex, the inner part is the medulla, and then this connects to the renal pelvis and the ureter, where basically the urine is actually conducted. And this distinction is very important, as you will see in a second, because these regions, the cortex and the medulla, have different roles. If we take a representation of a nephron here, like this juxtamedullary nephron, basically you have the glomerulus in the part of the cortex, the proximal tubule, the loop of Henle that's going deep down into the medulla, coming back the distal tubule, and finally ending in the collecting duct. The collecting duct will open in the renal pelvis down to the ureter. You also have some nephrons that do not go deep down into the medulla. Instead, they are much more superficial. What's important for our lecture today, I'm not going to be reviewing kidney physiology. This is something that you should have with you. But the important thing for this lecture is understanding how these long loops of Henle, these long nephrons, contribute to the possibility of concentrating the urine. And this is something that when you consider basically the osmotic pressure from the cortex to the inner part of the medulla, the observation that is inevitably to see is that the deeper you go into the medulla of the kidney, the higher the osmotic pressure. The deeper you go from the surface into the cortex, you have an osmotic pressure that is small, and then this keeps getting larger and larger as you go deep down in here. This is important because in order to extract water from the urine, in order to produce concentrated urine, you want to have the highest gradient possible in the renal medulla. And this is what this lecture is actually about. The longer the nephron, we could say, the longer the nephron, that means that the deeper the medulla, the larger is the urine concentration potential. The more you can concentrate the urine. And this is seen in this example here, even if what it's showing basically, this would be the part of the cortex, this is the medulla. And what you see is a progressive increase in the osmolarity in here from 300 milliosmoles that is present in the cortex. This goes down deep down to above 1000 milliosmoles. Why is this important? This is important because in the collecting duct you have the potential for recovering water. That is not always realized. In this situation here, for example, the wall or the epithelial cells in the collecting duct are actually, in this case, they are impermeable to water. What that means is that there is no water leaving here, and of course, in this case, you have actually a permeable collecting duct, which means that water is removed from the urinary filtrate here, or from the filtrate, is moved outside because of the gradient, of the osmotic gradient. The final result is that you have concentrated urine. If this wall of the collecting duct is impermeable to water, if this duct is impermeable to water, you are producing diluted urine, so this urine is not concentrated. What is it that makes the difference between an impermeable collecting duct or a permeable collecting duct? Is the presence, the stimulation of vasopressin. The hormone vasopressin basically turns this situation into this situation. When vasopressin is present, this collecting duct is highly permeable to water. Water can be removed from the filtrate back into the circulation, and actually you are producing highly concentrated urine. When we look at this problem from the perspective of differences, comparative physiology of different species, what we see is that the morphology of the kidney depends on the actual habitat of these species. Aquatic species look different from arid species. And the looks basically show that in aquatic animals, there is a much thinner, a much less developed renal medulla, so that the potential for reabsorption of water is much less. In arid species, in desert species, this renal medulla is more developed, allows for the existence of longer nephrons that will result in higher urine concentrations. Notice that this graph corresponds to humans. Humans are able to concentrate urine down to 1200 milliosmoles. If instead of humans, you take a desert species in which these loops of Henle are much longer, reaching almost down to the floor, the product would be that the concentration, the maximal osmotic concentration in the deepest side of the medulla is not 1000, but can be all the way to 6000 milliosmoles. And that's basically what you see in these arid species. The potential for actually having much higher urine concentrations. The potential for rescuing or reabsorbing much more water in the collecting duct. Finally, the potential for eliminating less water. And that's what a desert animal needs. Less to minimize the elimination of water. This is basically how the kidney in cross-section in a Mongolian gerbil and a sand rat or in a laboratory rat looks like. The extension excuse me, the extension of the renal pelvis is much larger in the sand rat. In the sand rat, basically you can have loops of Henle that are as long as this. This is an extremely long loop of Henle that will allow this animal to produce a very highly concentrated urine. Unlike in the common rat, where it can concentrate the urine much less. Basically, it's considered that a laboratory rat can concentrate the urine down up, still much more than humans, up to 3000 milliosmoles, but these animals can go down to 6000 and even 8000 milliosmoles. What if you look at what's called the relative medullary thickness, the thickness of the renal medulla, and you compare that between animals from different environments and habitats, arid environments in red versus, say, aquatic environments in blue, over a range of body masses, from very small animals to animals that are, well, 1 ton. What you see clearly here is that arid species always lie higher. That means that for a desert species, at a given body mass, the relative medullary thickness is always higher. The thickness of the renal medulla is higher. Just simply this. And if the relative medullary thickness is larger, the ability to concentrate the urine, the maximal urine concentration, also increases. Up to 6 or even 8000 up here. So, desert animals will have a thicker renal medulla that will allow for longer nephrons. Therefore, there is a higher concentration gradient, a higher osmotic gradient deep down into the medulla that will allow the animal to basically reabsorb more water and produce more concentrated urine. The basic mechanism is the same in humans as it is in the kangaroo rat, at least for all we know, and that is dependent on vasopressin. It is vasopressin that will maximize the urine concentration. But the potential is larger in the kangaroo rat in comparison to the lab rat, simply because the renal medullary thickness is much longer. this is the last study that I have been able to find that looked at this problem. And it's a comparison with modern techniques comparing the kidney of the kangaroo rat with the kidney of this is a normal laboratory rat. This is a nice study that has used the immunohistochemical techniques. And in these immunohistochemical techniques, they take a cross-section of the kidney, and in this case, this is deep down into the medulla. And what you see in blue here is the presence of aquaporin 2. Aquaporin 2 is the actual molecule that allows the increased permeation of water. Aquaporin 2 is stimulated by vasopressin. Notice basically the collecting tubules here, highly stained with aquaporin 2. That means that the high concentration of aquaporin 2 in the kangaroo rat, much less in the laboratory rat. You look at, in this case, a marker for the ascending thin limb of Henle. This is a chloride channel. Notice the nice ascending loops of Henle in opposition to the collecting ducts. Many more in the rat, much closer to the collecting duct in the kangaroo rat. And finally, aquaporin 1 positive, basically descending thin limbs of Henle in red here, in red here. What these authors have been able to do is basically by mapping the presence of these transporters in 3 dimensions, they give us a map of how the laboratory rat differs from the kangaroo rat, or the other way around. And their picture is the following. In a normal regular rat, what you see here is basically that the presence of the different markers, aquaporin 1 positive and negative in red, for example. The main difference is what you see here in red. Notice that in the kangaroo rat, it goes, the red parts that are aquaporin 1 positive go much deeper into the medulla as they do in the normal rat. What's the significance of that? Aquaporin 1 is a constitutive molecule that is responsible for the reabsorption of water in the proximal tubule and in the descending limb of Henle. The consequence here is that in kangaroo rats, because of the larger or the more presence of aquaporin 2 deeper down into the medulla, when the filtrate is going down the medulla, you can actually remove water from it for a longer portion of this of the medulla, the thickness, than you can actually do in the rat. As a consequence, you are removing, or the animal is removing, more water through aquaporin 1, therefore being able to concentrate the urine more. And that's basically the last information that we have about how this process occurs. So notice that in reality, this would be a nice adaptation for these animals. These animals in a desert environment make the medulla thicker, and they increase the expression of the transporters of aquaporin 1 deeper down into the medulla, enabled to reabsorb more water. This is not exclusive for rodents. It has been also shown for fish-eating bats. This bat is a fish-eating bat that lives in Mexico, and this is actually a comparison with 3 other bat species. Notice here, the chloride concentration in the urine increases progressively. The one that shows it highest is the animal that can concentrate the urine most. That is this species here, that is a fish-eating bat. That's the D picture. The D picture showing a larger relative medullary thickness in comparison with the other species, in particular with the C picture here, this bat. So, what's interesting here is that the same thing that appears in rodents, we see it in bats. Increased relative medullary thickness associated with an increased concentrating ability of the urine. It would be interesting to see if now we could put this in the perspective of the comparative phylogenetic method and look for basically traces or look for evidence of this being an adaptation. the increased relatively medullary thickness as an adaptation to arid environments. And this is how I wanted to finish this lecture. Thank you very much.